Hey, it's Mr. YS and this is a tutorial on 7.3 on translation. We've already had a look at the process of transcription, and in transcription we used the genetic information of DNA to transcribe, which meant we were creating a single strand. This is our single strand of mRNA. And that then leaves the nucleus and goes out into the cytoplasm. Now this is the version in eukaryotic cells which have a nucleus, so like our cells. But in bacteria, transcription and translation are happening in the same place. It's all within the cytosol. Although we do say the DNA, we can say it's in the nucleoid region. But it's not within a compartment. Okay, so what we're going to have a look at is the process of what happens once the single strand, which is over here, the single strand of mRNA has left the nucleus. And this shows kind of a free ribosome, but a lot of the time this will be happening in association with the rough endoplasmic reticulum. So the rough endoplasmic reticulum were those flattened sacs where we have ribosomes, so that we drew them as these circles, ribosomes associated with them. And actually what happens is the ribosomes attach to the rough endoplasmic reticulum and create the protein inside, and then that protein will move through before being packaged up into a vesicle. So we've looked at some of these ideas before, but never the details of what actually happens in the process of translation. How do we go from mRNA into making a polypeptide, which could then fold up into a protein? And here's a picture of the thing that's involved in making polypeptides. So the ribosome, you can see we've got different models of it. We're going to look at kind of a simplified model, and you can see it's kind of two sections. We've got this larger section here, and then we've got this small section here. And so we talk about, with ribosomes, the large subunit and the small subunit. And you can see in a molecular picture, it's a very complicated structure, and it involves both RNA and protein. And it's also an enzyme. So it's going to catalyze a reaction. And the reaction that it's going to catalyze, it's a anabolic reaction that we've talked about before, these kind of building up. Because it's going to be building up polypeptides. And what it's going to do, it's a condensation reaction that it's doing. In condensation, we remove water, and this is what was linking things. So it's a linking reaction. And just as a quick review, the thing that it's going to link are these structures. See if you can recognize what it is. It is nacho cheese, and nacho cheese over here, so it's an amino acid. So these are two amino acids, and the only thing that would be different would be their R group, and these two are going to be linked together by the ribosome. So what the ribosome does is it holds two amino acids close together, and in part of what is kind of the active site of it, it's going to remove that water. It will remove the water, and then we will get, so if these things are removed, the removal of water, then what we can do is we can now link those two together. And what we get forming is that peptide bond. So what a ribosome is a whole big structure to try and make peptide bonds to link together amino acids and to create a long chain of amino acids. Here's step one. So in step one of translation, The whole process is translation. And it's called translation because we're going from different languages. And so this is worth just stopping to have a think about. In mRNA, we have a language. If we zoomed in on the mRNA, we can see there's AUG written there. AUG, we're talking about adenine, uracil, and guanine. This is the language of nucleotides. So we saw the first process was transcription. We went from DNA and mRNA are both nucleotides. But in translation, we're going to go from nucleotides into the language of amino acids. And so that's why it's a translation. Okay, so in step one, what we see is that the small ribosomal subunit has come along and it binds, it's attaching to the mRNA. And we can say it the other way around, that the mRNA attaches to the small ribosomal subunit. So why does it do that? Well, because of the start codon, every translated mRNA will have this AUG, and this is almost, it's not quite a universal rule, but almost every translated mRNA will have AUG. So there are interactions there, and that means because of the structure of the ribosome, that AUG will bind to it. So the small ribosomal subunit and the mRNA interact, the mRNA binds to it. And you can see what's happened here is that we have had our first tRNA. So tRNA is transfer RNA. Our first transfer RNA has come up and it is binding. So we're seeing here a binding between the end of the tRNA and 3 bases on the mRNA. And this is what tRNA looks like more accurately. So on the diagrams above, you can see it kind of looks like a T shape as well. So transfer RNA. What is it? It's RNA, so this is RNA that is forming bonds with itself. It's kind of looping round and it forms this T kind of structure. And at the end of it, there is something, the acceptor stem, there's something where an amino acid can be tagged onto it. It can kind of tow, like it's attached, like cars attach and can tow things. Tow trucks can attach to cars and tow them along. It's able to attach to a particular, a specific, and we'll come back to this point, a specific amino acid. So there are different tRNAs which can attach to different specific amino acids. And what it's going to do is it's going to kind of taxi them in. So the tRNA is the transport mechanism for our amino acids. It's what's going to bring the right amino acid in, and we're going to see it's due to complementary base pairing. Here we see that the first tRNA, which we can call the initiator tRNA, is being coded for. So what it's coded for, what do we mean by that? Well, the mRNA, if we zoomed in on it, the mRNA had A, U, and G. They have that. And AUG is the start codon. So what is a codon? A codon is 3 bases. And we can talk about codons being on the DNA, the initial DNA, or here we're going to be talking about mRNA codons. So we're talking about 3 bases. So we could divide it up into 3 base sections, which gives the information for an amino acid. And so let's look what's happened here. There's AUG, and so if we look at complementary base pairing, then the matching ones will be A will match with a U, because remember there is no T in mRNA. There is no T in tRNA. There's no T in either of the RNAs. So we get an A matching with U matching with A, and the G matching with C. So due to complementary base pairing, a specific tRNA is the only one that can match. Only a tRNA that has this code at the end of it, the UAC, only if it's got UAC here will it match. If it's got a different code, it won't bond. There'll be no binding and it won't be able to bind there. But this particular one's being coded for, and at the end of that particular one, this particular transfer, there is always MET, which is actually the amino acid methionine. There is always methionine coded for. So if we call the 3 bases on the mRNA, if we call this the codon, the thing that is the kind of mirror image, we can imagine this kind of mirroring or the opposite. We've got the codon, and then the thing that's going to pair to it is the anticodon. So anticodons are on the end of tRNA, and codons are on mRNA, and they're going to match. So by a specific codon being matched by a specific anticodon, which is on the tRNA, we will get a specific amino acid being brought to the ribosome. So here's our summary of this. It can be called initiation because it's how we start. It's the first thing that happens. So in initiation, the mRNA binds to the small ribosomal subunit. The initiated tRNA, which is always MET, we can call it MET tRNA because it's a methionine attached to that tRNA, will bind to the start codon AUG, which is on the mRNA. Now this tRNA, the tRNA has an anticodon which is complementary to the codon on the mRNA. And each tRNA has a specific amino acid attached to it. In this case, it was methionine. But we have 20 different amino acids that we could be giving the code for on the codon, on the mRNA. The next step is then that the large ribosomal subunit comes in and it binds, and now we've got the complete ribosome. And you can see that there are some different sites that we've labeled here. There are 3 sites of activity. The E site, which is the exit site, the P site, which is the peptidyl, to do with the idea of the peptide bond, and the A site, which is the amino acyl site. Now these, we can abbreviate them to EPA. So you could ignore these words, but you might see them or you might wonder what they are. We do call it the exit site for the E. Okay, so we can see once the initiator MET tRNA has bound, and it was due to that complementary bases on these, sorry, other way around. It's going to be It's AUG here and UAC there. Once it's bound and the large ribosomal subunit has attached, we can see that the initiator tRNA is in the P site. So it doesn't bind in this first site here. We're going to kind of see that this, even though we're labeling it EPA, we kind of think of them as being almost 1, 2, 3. So the initiator tRNA binds in the second site, which is the P site. And our summary is that the initiator tRNA is in the P site and the large ribosomal subunit binds to the small. Then we have the next tRNA will come along. So here's another tRNA, and it is also going to have an anticodon on it. And that anticodon, so we had our first codon there, codon number 1. And then if we read that way, we've got the second codon on the mRNA, which is going to be 3 bases. And that second codon will match 3 bases on another tRNA, the anticodon, and this will have another amino acid, another specific amino acid at the back. If we think about this with lettering everything, we can see that the first one, AUG, that did complementary base pairing with UAC to bring in methionine. Now, if the next codon along was CAC, that would match with GUG on this tRNA. And the tRNA with the anticodon GUG always attaches to HIS, which is histidine. And so this is where our translation's occurring. By matching these two together, we get a particular tRNA which has a specific amino acid on it. And so what the ribosome does is it just reads 3 at a time, every 3 bases on the mRNA. It will then hold that in place and allow the correct one, whatever it is, to come in. We get another tRNA bonding and another. It will allow the correct tRNA with complementary base pairing to come and bind. So we can summarize this by saying the next codon on the mRNA signals for the next tRNA to come in, which will bind, and we can see the next one is going to come in and fill this slot here, which is the A site. So it binds to the empty A site. Now the ribosome has read 2 codons. We've read the first 2 codons, and the first 2 tRNAs have come in and they've bound due to complementary base pairing. And now, because they are bringing with them MET, and let's say this one was HIS, we've now got 2 amino acids close together. And this is where this bit up here, that's the kind of enzyme, that's the enzyme part of the ribosome. So now what can happen is it can link those 2 together. It can do the condensation reaction to link these 2 amino acids together. We can summarize this by saying a peptide bond forms between the amino acids which are still attached to the tRNAs. So between the amino acids of the tRNAs which are in the P and the A site. So it's between these first 2. The A and the P are the ones which are going to do the peptide bond formation. The tRNAs which are in the A and the P, the peptide bond formation is between those 2. Next, let's see if you can work out what's happened here then. This was the original MET tRNA, and you can see it's missing its amino acid because the met is now here. So what's happened? Well, the peptide bond had formed between our first two amino acids. The ribosome has moved along, it's moved up 3, because now you can see that our second amino acid has moved from the A to the P site, and our met, our green one, which was in the middle site, it's now moved and it's actually left out of the E site. It's left from the exit site. So it's moved up 3. It has broken the bond which was attaching the met tRNA to the methionine. So we can see that this is now coming away and this is empty. This tRNA now doesn't have its amino acid, and it's transferred that amino acid, because it's now peptide bond, it's transferred this to the amino acid which is now in the P site. So we can see that the one that's now in the P site, this is the one that's going to hold what we call the growing polypeptide chain. Because each time what was in the A will move to the P, what was in the P will move to the E and then leave, and the one which is in the P will be left holding all the amino acids. Now we're going to see when another one comes in here, then we will transfer that chain on through the peptide bond to the one that comes and binds in here to the A site. So the P site is where we hold the growing polypeptide chain. So we're going A to the P for our one that was first in the A site, P to the E, and E out of the ribosome. And this, because it's moving along, this is called translocation. The idea of moving, the ribosomes moving along the mRNA. So here's our summary. This, because we're growing the chain, is we can call elongation, and it's a series of repeated steps. The ribosome translocates 3 bases along the mRNA. The tRNA, which was in the A site, moves to the P site. The tRNA, which was in the P site, moves to the E site and is released. So it's really important that we're talking about the movement. It's the tRNAs which are moved between these, and it's the amino acids which are attached at the back. And so then once one amino acid has left, the first one which was in the P site has now moved to the E and left. The one which was in the A site has moved to the P site. We now have an empty A site. And so again, reading 3 bases at a time, we will get those complementary bases matching, the G matching with the C, the U matching with the A. It will come in and it will bind. So again, we get another anticodon, the next complementary anticodon on the specific tRNA will come and bind to the A site, bringing along the specific amino acid. And then we're going to repeat. So it's a series of repeated steps. As the new one comes in and binds, we then make a new peptide bond. In this step here, we can see that they're linked. It then moves along, it translocates, releasing one and then freeing up the space for a new one to come in, and it just continues to do these 3 base movements, adding in a new tRNA to the A site, making the peptide bond, moving along, releasing from the E site, and it just continues to repeat. So a quick summary of this picture up here was that whenever it's moved along, we get a new tRNA with an anticodon complementary to the codon on the mRNA binding to the vacant A site. And it keeps moving along, extending this polypeptide, the growing polypeptide chain, which again we can see is being held by the P site, until eventually we get to a stop codon, e.g. UAG, and translation is terminated when a stop codon is reached. What happens is when we reach a stop codon, this is called a release factor. So it's not a tRNA. Something which isn't a tRNA that's called a release factor comes and binds into the A site, and that can't make a peptide bond. And this causes, let's see if we can work out what's happened here. Well, first of all, the small, the large, and the small have separated. The fancy way of saying that is dissociated. So they have the large and small subunits have dissociated or separated. We can see that the mRNA that was used has been released, and we've also released the free polypeptide. We can summarize that with the small and the large ribosomal subunits separate. The mRNA separates or is released, it detaches, is released. So is the polypeptide, and then we can fold the polypeptide into a protein. Okay, all of these final steps were, because it's the end, the final steps can be called termination. And that's it.